Hello everyone, my name is Chase Bernardi. Today's date is September 9th, 2025. Here's my paper. And today for Cell Bio Week 3, Mod 4, today we are going over how cells produce and transport three types of macromolecules: a lipid hormone, a secreted protein, and intracellular protein. So we have our diagram. Each macromolecule follows a unique pathway dependent on its chemistry and function. Lipids are produced in the smooth endoplasmic reticulum, proteins for export move through the rough endoplasmic reticulum and the Golgi apparatus, and finally intracellular proteins remain in the cytoplasm. For our lipid example, which is cortisol at the bottom, cortisol is a steroid hormone synthesized in the smooth endoplasmic reticulum. Starts off as cholesterol, is then modified by an enzyme to form cortisol. Because it is lipid soluble, it can cross directly through the plasma membrane without vesicles and enters the bloodstream bound to carrier proteins. For our secreted protein, we are going to talk about the growth hormone. So growth hormone is synthesized on ribosomes bound to the rough endoplasmic reticulum. This is going to be the second or middle chart. Inside the endoplasmic reticulum, it folds and it is glycosylated. It is then transported to vesicles carrying it to the Golgi apparatus, where it is processed and packaged. It is then stored in secretory vesicles and released by exocytosis into the bloodstream. Our final example, and the top example, is intracellular proteins, which is actin. So actin is synthesized by free ribosomes in the cytoplasm. After folding, actin monomers assemble into filaments that form cytoskeleton. This supports a cell, enables mobility, and powers intracellular transport. This flowchart summarizes the differences. Cortisol diffuses out. Growth hormone uses the endoplasmic reticulum Golgi vesicle pathway, and actin stays inside the cytoplasm. In summary, the cell directs molecules based on their function. Steroid lipids cross membranes freely, secreted proteins follow the endomembrane system, and intracellular proteins remain where they are needed inside the cell. References are attached to my final documentation. Thank you so much, and if you need any questions, please reach out to me and have a good day.